Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 2, Episode 4 of Surfing the NASH Tsunami starts now. First, for those of you who sent private notes thanking us for focusing on an MLK Day-related theme last week, it was our pleasure. I believe Stephen may be able to talk about, I know he and Don have been in touch on some of the things we discussed during that episode. We are sending the second conversation out to some folks that we think might have interest in moving forward with it. And if you want to help us realize what we were talking about in terms of education at vaccine centers, please let us know through questions tab on the website or sending an email to surfingnash.com. Number 2, in our never-ending growth curve, we are now averaging over 1,500 30-day downloads in the past week. Thanks for your continued support and interest. Speaking of support and interest, we intend to keep the music contest up one more week, and we'll be sending one more set of notes about it. I want to thank Stephen, a listener, commending us on all 4 choices. If you've heard them, there are 4 different takes on what surfing music might be. I think you'll want to vote on this because you're going to be hearing the winner every week, at least once, maybe twice. So if you want to get a vote on what you're listening to, I think that's a good thing. And with that, let's go on to the podcast. This is going to be an exciting week for this podcast. On Friday, FDA is holding a long-anticipated meeting titled Regulatory Perspectives for the Development of Drugs for the Treatment of NASH, focusing on clinical trials, phase 2, phase 3, endpoints, a whole range of the issues we've discussed. And this long-awaited event is likely to stem from a piece that was recently published in Hepatology titled Non-Alcoholic Steatohepatitis, Current Thinking from the Division of Hepatology and Nutrition of Food and Drug Administration. So today, we're going to discuss some questions and expectations about this meeting. And our good friend Manal Abdelmalek is joining us here. And then right after the webcast on Friday, we will take another session, including the 4 of us, as well as an industry executive at Yale, the Chief Development Officer at Akero, will be joining us. And with that, let's get started. One good thing that happened in the past week, let's do professional. Although sometimes, as I said, let's do professional is personal. Brave one first.
Oh, I'll jump in then. I suppose my professional this week is just the planning that we've got going on for post-COVID recovery to assist in getting FibroScan lists back up and running, get hepatologists, hepatology patients through to see hepatologists in a timely manner, and the various different ways that we can assist in doing that. So we're just in planning phases for that because we don't know when we're going to be able to assist trusts and primary care or even clinical trials in that at the moment. But there's a lot more interest in trying to streamline processes and get things back up and running, which is encouraging.
Well, thanks, Louise. That's a great way to start us off.
I'll jump in. First of all, I forgot to, you can cut this from the tape, but I haven't seen this view from Manal's, camera yet. So I've never seen the plant and, the curtains. I don't know if we're actually in a bedroom there or I hate to ask where other places that might be.
Yeah, inquiring minds want to know.
Manal, I would say a woman of mystery and not say a word.
I like that position. Just a different part of my house, Stephen.
I love it.
Yeah. Kids are in the family room. So study's taken, the table's taken. Had to find a quaint corner somewhere else.
I love it. Good. Well, I actually just have a professional update. So since we talked last Monday, we actually made some pretty good inroads on our first attempt at disease state awareness and education to our COVID vaccination patients and citizens of San Antonio. So we were able to go out to the Alamo Dome and actually speak to patients as they were leaving the vaccination center. And we were able to talk to approximately 60 individuals about fatty liver, and it was incredibly well received. We were able to hand out educational materials, in part put together by the Global Liver Institute, as well as Summit Clinical Research, really just educational awareness more than anything else. And if they wanted to know more, we linked them to an opportunity to get a FibroScan. I do not have results on how many of those patients actually, or how many of those people actually followed up, but really it was more about how was it received. And everybody to a person really appreciated the educational material given to them. Most of them said they had never heard of it. As a kind of a mini pilot, it was very informative. Next steps are to really work with the city of San Antonio and other constituents of the city to broaden out the awareness campaign. And that's really all it is. It's just bringing education to patients as they are in line to get a vaccine or as they wait for the requisite 15 minutes before they can leave to make sure they don't have a reaction. And my hope and goal is if we increase awareness of this disease by even 10%, I think that would be huge. So a good start, I think.
Good job. Manal, I guess that leaves you.
Well, that's a hard one to follow, Stephen. That's pretty cool. My big professional is I was really excited to see a large rollout of an invitation for other personnel to start helping with the COVID vaccine broadly, whether it be faculty or medical students or LPNs or anybody that was eager to help with the vaccinations. And my high point this past week was really the and that I've received from patients. I was doing liver biopsies, and my chagrin, I had one patient who has already been in repeated clinical trials and was here for her liver biopsy over the past 10 years of my career. And I was aghast saying, wow. And she said, I know we're getting closer to finding it. And I have to tell you, overall, I feel just so much better doing this mission. And despite the negative trial outcomes that have happened for which you indicated, to see patients invested in this was really a high point of my week, especially that type of enthusiasm, despite the interventional nature of the endpoint.
Thanks, Manal. That's great. And that enthusiasm really that is the point. A lot of this now is about getting people engaged and keeping them engaged. So I didn't mention this at the outset. I'm back in Corpus Christi for my granddaughter's birthday party last weekend. But that means, while I've got some great personal moments, not a lot of professional bets. I did have a client that I made a recommendation to, not in the liver space elsewhere, who got feedback from the agency this week that suggested that my recommendation, which they thought was wildly optimistic and off base, might turn out to be closer to reality. So that was a good thing. With that, let's go on to our event. We're just going to spend the rest of the meeting answering a few questions developed in advance about expectations for Friday's session. At each round, each of us is going to answer, and then we'll talk for a couple of minutes, and we'll go on. Kitty Yale, who'll be with us on Friday, contributed some answers to one of these questions, which I will read when the time is right. But so on the first round, we're going to start with different people on each round. First round, let's start with Stephen. Stephen, what overall guidance do you hope we will receive in the FDA for Friday's webcast?
Thanks, Roger. First of all, I have to say I'm excited to have the FDA put together this open forum discussion on where they stand with NASH clinical trials. I do think you can never communicate too much. So the more they communicate their stance, I think it becomes more clear to those of us who spend our life focused on drug development in the field of NASH. And I think we can get a little bit of insight into what their thinking is by reading the recent white paper that was published in Hepatology. You can access it through the Hepatology website, but it's not out in hard print yet. And a couple things in there that I'm hoping that they provide some clarity on. Early on, there was a discussion when the Intercept drug, obeticholic acid, received a complete response letter that maybe the goalpost had moved. And I think what was inferred there is maybe the endpoints had changed. And I think just understanding the formal white paper written by the FDA, those goalposts, to my knowledge, have not changed. The Subpart H surrogate endpoint remains as stated. Resolution of NASH without worsening of fibrosis, improvement of fibrosis by at least one stage without worsening of NASH, or hitting both combined endpoints. And I just want clarity from them that is indeed still the case. I do find it interesting, however, in maybe the first couple paragraphs of the white paper, they make it clear that NASH with liver fibrosis is a serious life-threatening condition and as such deserves drugs to treat it. They also go on to say the magnitude of the benefit a patient receives with lifelong treatment must be balanced with the safety profile of the drug. What that brings to my mind is I want to hear from the agency kind of where that priority is, right? So is it, to me, based on the CRL that we know was sent to Intercept, the wording that was translated without having ever seen the CRL, what was conveyed was that there was an inadequate risk-benefit ratio, or there was a concern about a risk-benefit ratio. And when you read this white paper, it's pretty clear that safety is on the minds of the FDA in a big way. Again, repeating, the magnitude of the benefit a patient receives with lifelong treatment of NASH must be balanced with the safety profile of the drug. NASH patients are also vulnerable to other diseases, and investigational drugs should not worsen comorbidities, including cardiovascular disease, hyperlipidemia, metabolic disease, and diabetes, or cause liver injury. And so I hope that they just provide a little clarity around that. The other thing I would like to learn a little bit more about that I don't quite fully get a clear picture of from the white paper is their stance on non-invasive biomarkers and what it will take working with the biomarker qualification program within the FDA to get to where we're acceptable, or they are acceptable, of a non-invasive test acting as a surrogate of a surrogate for an outcome measure. My guess is somehow that's going to work through machine learning and artificial intelligence, where we're looking at fully quantitative assessment of the NAS lesions, NASH resolution per se, and then also fibrosis improvement. But it's unclear to me exactly how that would work out, and I'm hopeful that there is some clarity provided on Friday as well. Also, would be nice to get a little stance on this idea of placebo response and how pathology is interpreted in the era of doing liver biopsies. And we've talked on the podcast quite a bit about inter- and intra-observer variability, the issues with liver biopsy interpretation, and they go into that a bit in their paper. And they do provide some rough outline there about recommending sponsors review in detail their plan for liver biopsy procurement, processing, and interpretation. And I get this quite a bit from sponsors. They want to know what that means. And there is a little bit of guidance in here, but I'm hoping it's fleshed out a bit more in the formal review that's going to be public on Friday. In other words, do we need to have two pathologists? And what they do mention is that because there's a high degree of discordance in liver histology interpretation, that maybe that could be improved by pathologist training. For example, having an adjudication committee of central pathologists read baseline and treatment slides together and decide how each of the components of the NAS system would be interpreted, with the notion that such training efforts should help address many of the limitations of histopathology reading. So really, just some more clarity around what that means. And I think with that, I'll stop.
So Stephen, first, I just want to note that a lot of what you just mentioned aligns with some of the comments you made on the year-end summary episode about what the agency had indicated up to that point. And there, you talked about the kind of statements that you now want to get a lot more detail on, and I think that's a good flow, and it all made a lot of sense then and makes a lot of sense now. Let me turn next to Manal and ask the same question.
Yeah, I couldn't agree more with Stephen's points. I think we do need some guidance as to how do we balance in the context of executing effectively clinical research with a safety efficacy balance. There is a little bit of a difference in clinical practice versus clinical research. Side effects related to drug therapies can sometimes be mitigated, for example, with dose reduction or concomitant therapies. And it would be wonderful to receive guidance as we develop these clinical trial protocols, how we can ensure the highest level of safety in the context of the trials, such that when we get to achieving efficacy endpoints, this unknown balance of long-term safety versus efficacy isn't so much in question. Do we need to implement risk mitigation strategies in the context of clinical trial design? Should we leave risk mitigation up to a manner in which a provider would otherwise manage a lipid increase or a glucose signal or any event that would come up in the context of a clinical study in the way that they would otherwise be at liberties to manage it had they otherwise been in in clinical practice. So I think a guidance there would be, highly welcomed. I would also like to see where the industry is going with their thoughts about how we're going to utilize the totality of data. We've certainly come to a further understanding of the imperfections of liver biopsy on multiple fronts that are even beyond pathology interpretation alone. Sampling variances, sample quality, and needle size. Standardizing that in phase 3 clinical trials is fairly onerous, let alone whether we use one pathologist to committee review, blinded pre and posts, restaggering the deck and rereading them at the end of the trial. I think that can have really a lot of bearing on an outcome or even a change in the outcome. And are we at a point now, given the ability to track change with surrogate markers, whether they be imaging-based or biopsy-based, for the emphasis on biopsy to be looked at in part, but not in its totality as a primary endpoint, maybe as a conglomerate endpoint with other surrogates, and depending on the strength of the surrogates, is it all going to hinge eventually on that fibrosis stage on biopsy, or maybe even a replacement of these surrogates? I think we have to move in a different direction to redefine, quote, a different gold standard, because it is disincentivizing our ability and I think hindering the pace for drug development and getting promising drugs to a large population at risk.
Thanks, Manal. Just wondering, was there anything you read in the document that led you to think they might go so far as to talk about biopsy as part of a composite endpoint, or is that on the wish list?
That's a wish list, I think. Ultimately, we will be treating towards harder outcomes, feel, function, and survive. And a biopsy change from an F3 to an F4 doesn't necessarily change our clinical practice, and understanding the misclassification rate even between those two stages on biopsy, it makes me wonder, are we putting too much emphasis on such an imperfect endpoint in deciding how we propel promising therapies forward if other surrogates are supportive of a positive change from baseline?
It's interesting. I'd highlighted some of the similar parts that Stephen was talking about, and I totally agree with Manal. And I think I looked more at the beginning of some of the documents where they're actively encouraging the movement towards the non-invasive testing and the biomarkers and the proof of concept studies now and the phase 2. And I think that certainly opens clinical trials up to the non-normal clinical trials units. I think patients do prefer non-invasive techniques, and I think if we can do a lot of the work with non-invasive techniques and biomarkers, I think that allows us to strengthen potentially the field of non-invasive biomarkers that we're looking at. We obviously have ELF-NIS for NAFLD scores. We have all of those that are working on MRI dynamic fat fraction. We've got FibroScan. We now have Smart Exam. Things are moving in that direction. My only concern is if we do use these a lot more in proof of concept studies and the early phases, the documents are fairly clear that when we move to the phase 3 studies, it's histopathology, and it's the changes in histopathology that we need to see, as Stephen so rightly said about the end points. But how do we then move from a more non-invasive early assessments into phase 3 with the accuracy that we're going to need? So there was a lot of encouragement, and I'd certainly like to see some clarity in exactly how we're going to transcend that barrier across from the phase 2 to the phase 3 studies, where we change from non-invasive to histopathology as the primary method of review. But I do think it's encouraging. I think we'll see a lot more patients able to be recruited. We may well get closer to finding out the natural history of fatty liver disease, because I think we don't know who's going to be most affected, and we can't pick those people realistically. But we're not doing enough early screening to find the people that we can monitor on a long-term basis. So hopefully by further developments of non-invasive testing and biomarkers, we can strengthen how we can diagnose liver disease, because currently we know they're not the best. So those are the bits that I was really interested in, was that movement. And I suppose it takes to Donna's and the GLI that maybe we are moving a little bit more beyond the biopsy in the early start of the clinical trials.
Thanks, Louise. Let me just share mine and then throw it open for questions and comments between us on this topic before we go on to the next question. There was a section in the middle of page 5, I also highlighted, that talked about sample sizes. NASH is a common disease. Trials that provide a sufficiently large pre-approval safety database will facilitate assessment of risk and benefit. What they then asked for was the size of a pre-approval safety database that would ensure that low-frequency adverse events could be detected and appropriately described for assessment of risk-benefit, and went on to tell sponsors they should be aware that the size of a placebo-controlled trial adequately powered for efficacy might not be sufficient to support the drug's safety, allow for the overall benefit-risk assessment that's necessary for drug approval. To the degree that that simply says larger samples, I guess that's one thing, but it could say larger samples, it could say longer studies, it could say you're going to need to find a way to aggregate data from multiple sources to make this make sense. It could say 2 trials. What that paragraph winds up meaning, I think, will have a tremendous practical impact on the cost of getting to market and the time it takes to get to market. Therefore, exactly how high the hurdle for clinical success in an economic sense is going to wind up being. So that would be mine. And anybody have questions, comments for any of the others of us on any of this?
If a drug is highly efficacious and has a very clean safety profile, I think the number of data points that the FDA or any safety review committee needs to look at is finite. However, where you have drugs that have a potential safety risk, as highlighted in phase 2 or maybe as part of your NDA filing, and the efficacy is relatively marginal, then I do think that you may be held to a higher number of patients to provide that bit of further sense that you've done your due diligence. And I think that's been part of the challenge so far. Beta-cholic acid, we use it for primary biliary cholangitis. It's been around now in the market for a little while. It's been extensively studied in large numbers of patients with NASH, and there are some issues related to the drug, and the overall efficacy is relatively marginal. 11% placebo-adjusted response rate on fibrosis, while highly significant. The clinical significance of that is good if you happen to be in the group that's having a fibrosis benefit. But in the rest, where you're not having a benefit, are you faced with some side effects or adverse events that maybe are unwieldy? Maybe it's high elevated LDL cholesterol, pruritus, gallstones. I don't know. But I think that's the concern, is they just want to have a very clear picture of, what's happening to the patient from a safety perspective relative to the efficacy. And I think, to your point, it would be nice to hear them spell that out with a bit of granularity and clarity, rather than just saying the risk-benefit ratio has to be at a certain level for us to have a comfort with it. Tell us what that is. What do we need to show?
I agree with your question wholeheartedly. The source of my concern is that there's a different way to interpret that paragraph in the context of diseases that affect millions of people, which is that you start looking for 1 in 1,000 or 1 in 10,000 effects. And when they use the phrase, which I'll pull up again, low frequency adverse event, I don't know what that, and that's a phrase I would love to have significant clarity on, because if they mean 1% of the population, then I think your description holds. If they mean 1 in 10,000, as they do in some other drug classes, that creates a whole different set of pressure on drug trials. So I just think we need to be clear on what it is. I don't think it's going to be what I fear, but I think clarity would be really helpful.
I suppose I just had a slight question that followed on from yours, because after the section that you were discussing, they go on to sort of state that clinical trials might want to look at an endpoint, a histopathologic endpoint at 2 years because of the slowness and the subtle changes with the disease. So that's moving it from the 12 to 18 months, which is current. So when Stephen was saying earlier about we don't know whether they want longer studies or bigger studies, it seems like they're sort of lengthening the time points for some of these biopsies. use, but also post-market setting, reconsenting patients to a drug trial when the drug's already been approved. Is that what I was reading in that document? So it just didn't sit comfortably when you have patients in a phase 4 post-marketing study, then you've got a drug that's approved, to have to then reconsent them and try and keep them in that pathway after the drug's available. And I suppose that I just wasn't clear on where they were really getting to and what they wanted us to do specifically with patients at those time points. I can read the section. The sponsors might want to consider efficacy evaluations of the surrogate histopathologic endpoint at 2 or more years because of the subtle changes in the histologic features associated with the disease. Moreover, trials should be designed to follow patients who continue their randomized treatment assignments, e.g. investigational drug or placebo, in a double-blinded fashion for clinical outcome assessments in the post-market setting to be able to confirm clinical benefit following accelerated approval. There are obvious challenges associated with this approach. For example, reconsenting patients enrolled in the trials to ensure awareness of the availability of an FDA-approved drug under accelerated approval. So that, to me, was reconsenting people on a placebo line when they could actually be receiving the drug that had been approved by accelerated approval. Now, I don't know how comfortable the clinical sponsors will be with that or how facilitating a patient would be with that. I wasn't quite clear on that.
Yeah, well, Louise, that's the way it's currently done. Right now, we have Subpart H approval, right? So you take 1 or 2 doses of drug plus a placebo, you randomize the patients, they get treated for 1 year to 18 months, repeat a liver biopsy, see if you meet the Subpart H histopathological endpoint. If you do, you submit your NDA and you may or may not get approved. But at the same time, if you read further down on that, or into the next paragraph, it talks about you must begin your phase 4 portion, or the long-term outcome portion of your phase 3 has to begin prior to the presentation of the Subpart H database, filing of your NDA. So before you submit your Subpart H results, you have to be underway with the long-term outcome portion, which is a blinded portion. So Intercept still doing their phase 4 portion right now. That's the long-term outcome piece. So those patients, we still have patients that are on IP and some are on placebo. And the idea there is you go until an adjudication committee is able to evaluate a certain number of events and show that significantly more have occurred in the placebo arm. And, for most studies designed so far, it's somewhere between, roughly around 400 to 450 adjudicated events. And they outline in that white paper what those adjudicated events are. And they will probably, I suspect, go through those again on the call Friday, which are essentially progression to cirrhosis, which, quite frankly, if you're in a non-cirrhotic trial, an F2, F3 study, my own opinion is progression to cirrhosis will be the number one adjudicated event. Reduction in hepatic decompensation, meaning lesser numbers of people on drug having variceal bleeding, ascites, hepatic encephalopathy, fewer people progressing from a MELD less than 12 to a MELD greater than or equal to 15, fewer people dying from disease of whatever etiology, and fewer people having liver transplantation. Now, you did raise a very interesting question about the document, and that was their wording that suggested maybe trials should be 2 years in duration. Given the slow, gradual progression or improvement in inflammatory changes in fibrosis observed on liver histopathology, sponsors might want to consider efficacy evaluations of the surrogate histopathologic endpoint at 2 or more years. Now, that's fine for drugs that are slow to work, maybe have a marginal benefit. But I'm here to tell you that this treatment paradigm is rapidly evolving in the field of NASH. The pharmaceutical industry has gotten really good at targeting the pathological principles, the pathophysiological principles of NASH. And these early paired liver biopsy studies are showing some real positive impacts on NASH in fibrosis at relatively early time points, certainly much earlier than 2 years. So I think while, look, way back when we started this in the Pivens trial with vitamin E and pioglitazone, it was a 2-year study. And then we did, 18 months for Intercept. And I just think that our drugs are getting more and more efficacious. They're targeting the real drivers of NASH and fibrosis. And quite frankly, some of our drugs are defatting the liver at a very rapid pace. And I think we will begin to see that equates to improvement on histopathology at a much earlier time point than 2 years. So I'm hopeful that there is some clarity provided there, or at least, if not clarity, a willingness to say, well, look at the phase 2 data, and we will listen to the sponsors outline their phase 3 protocol and plan. And if the data warrant, then potentially you wouldn't need that long of a phase 3 trial. I think 1 year for some of the drugs that are in the pipeline is significantly adequate to show a dramatic response on liver biopsy. I just think they need to clarify that statement a little bit more.
Thanks, Stephen. Let's jump on to the next question. Manal, are there 2 or 3 specific items you'll be listening for, and if so, what are they?
I think, as we discussed, the totality of data, even if it's a surrogate data as it pertains to endpoints, is going to be very important. The other thing I'm going to be interested in looking for, because all these issues that we're talking about, safety, the balance with efficacy, the determination of the ideal efficacy endpoint, which may be in part uniquely different based on the target which is being evaluated. It certainly is not one size fits all, whether it be in duration of study to time of endpoint, or potentially even the endpoint itself. We've seen anti-NASH drugs that may not hit fibrosis at a unique time point, and fibrosis be met without resolution of NASH. So I really do think that this is going to have to be appraised on a unique target, unique sponsor basis and a comfort level. But this all still feeds into some undercurrents which we haven't touched on. Our screen fail rate for patients who on clinical premises have NASH and for all practical purposes elevated liver enzymes or fat on ultrasound, failing to be included in studies because they didn't meet the definite NASH histologic endpoint, and screen fail rates that go anywhere between as high as 40 to 70% in trials. But also the depth of heterogeneity of patients that are included into clinical trials because they have the histological endpoint, but yet be clinically very heterogeneous. Diabetics and non-diabetics, dyslipidemics and non-dyslipidemics, obese and lean patients, all kind of going into comparable trials. And we have not yet mastered appropriately phenotyping our patients using endpoints other than just histologic. I would be very interested to see what the agency has to say about our patient selection and insights on the screen fail rates and potentially how to optimize that such that we can be more targeted in our ability to interpret the safety efficacy endpoints and potentially even optimize them beyond what we're seeing. We may be diluting out our ability to achieve a registration endpoint because of the complexity and the heterogeneity of the disease state itself.
Great comments all. Any clarifying questions for Manal, and then we can go on.
Well, let me just say I completely support and agree with what Manal said, and maybe I'll extend it a little bit more and say, look, in our phase 2 program, do we even need to be doing liver biopsy to qualify for the study? And can we begin to take the data that we have in front of us and that we've presented and published and use it to define you can call it a presumed NASH population or a probable NASH, or I would like to just call it a NASH population using non-invasive tests. Maybe that's MRI-PDFF to define the fat content in a non-cirrhotic trial, and then MR elastography or a FibroScan to help define the amount of fibrosis. I'm a little leery of FibroScan because of its positive predictive value not being as high. But I do think we can we can get at a presumed NASH population for phase 2 without doing liver biopsy by looking at something as simple as an AST above a certain threshold. You have fat, we quantify that, and then we can get at some metric of liver stiffness. And we're getting really good now at some of our clinical tools. In fact, Rohit Lumba published a paper I think it came out online this month in Gut, showing that a FIB4 of 1.6 with an MRE of 3.2 or 3.3 actually had a positive predictive value of something like 80% for NASH, and that could be used as basically a tool to define the NASH population. That would help us significantly, Manal, in enrolling our patients and getting away from some of this heterogeneity and sampling variability and intra-observer variability statistic we have. And quite frankly, if we don't do a biopsy on the front end, well, do we really need one on the back end? Let's just repeat those non-invasive tests, and then we learn from that. And yes, while we still need to do phase 3 liver biopsies, because that's our subpart H surrogate endpoint, I think we can all agree, or hopefully we can reach consensus, that you are de-risking your phase 3 by showing very positive results on non-invasive tests in phase 2. And I think that would encourage enrollment. It would speed up enrollment timelines. And I'm not sure it would hurt us at all getting into phase 3, where you still have to have that biopsy at the beginning and the end. I threw that out there maybe as a little bit of a provocative statement, but I think it would help overall.
I don't think it's a provocative statement. And I think if you look at the draft guidance, they've actually left open the door to answer Manal's question. In the phase 2 development considerations, they've got a nice paragraph in there, which in the early trials, baseline histological documentation of NASH may not always be needed, depending on the endpoints to be assessed. Sponsors can enroll patients based on either known histological diagnosis or NASH or a combination of biochemical criteria and or imaging evidence of steatosis, steatohepatitis, fibrosis, in addition to known risk factors for NASH. So I think in their development considerations, they're really nicely asking us to look at non-invasive ways to categorize the patients that both you and Manal have just described, and to basically invent your own studies with those criteria and to prove them as clinically accurate as we can. And I think that's great.
I agree with you. The problem is what's not clear is that a 2A or a 2B? Because very historically, very classically, we do 2A. We've gotten to the point where in 2As, we just do non-invasive testing, but it's in smaller numbers of patients. It's proof of concept. Are we moving the needle? And does that warrant then a more expensive liver biopsy phase 2B, where we show histopathology improving, and then we use that to power our phase 3 based on the magnitude of effect change that we see? And it also helps trigger which endpoint we're going to go after. In other words, are we seeing more NASH resolution? Are we seeing more fibrosis benefit? And so that helps decide, okay, where do we want to spend our alpha? And I think if they could give us some clarity around that, Louise, saying, look, even in phase 2B, you can come to your end of phase 2B meeting with the FDA with non-invasive data. That would be, in fact, I might even ask that question if I get the opportunity, because I think that would be provocative. That would be provocative.
I totally agree. A couple of things. First of all, you could just bulk up your 2As. If that was the place you could go. I understand it doesn't solve the problem in 2B. But second, I want to read Kitty's questions, because one of them in particular goes back to the downside of all this, which stems from, I think, the question I asked about what you need for sample size. Kitty had said that she was looking for 4 specific. One, clarity around their statement regarding worsening comorbidities. Quoting here, NASH patients are also vulnerable to other diseases, and the investigational drug should not worsen comorbidities, including cardiovascular disease, hyperlipidemia, metabolic disease, diabetes, or cause liver injury. Her comment was, this has been a challenge for a number of MOAs and provides clear direction for expectations to the next generation of drugs in development. I think that's a focused statement more than a question per se. Number 2, additional guidance on size of overall pre-approval safety database. And there the quote she used is provide a sufficiently large pre-approval safety database that will facilitate the assessment of risk and benefit. And what she was asking is, they talk about the ICH guidelines. The ICH guidelines would suggest larger than 1,500 samples. And that would align with my comment about they're looking for 1 in 1,000 events. And if, in fact, that's what they're looking for, and all that has to come in phase 3, particularly given that they've noted as well that you might need more than one pivotal study. I'm not sure how all these pieces fit together, but that one jumped out at me, and then it jumped out at her as well for the same reason. And it's kind of the flip of what we're talking about here. Although if you could get to the end of 2B without having done biopsy, and we're able to put enough patients into it that you had better and deeper information, how would that play out against any of these sample size issues or phase 3 issues, as she noted? And then her third comment, FDA have provided clear guidance on requirements of histological analysis in terms of inter-reader concordance and should lead to standardization across studies. Great to see the process outlined in the manuscript aligns with our recent FDA feedback for upcoming studies, which I read as saying she sees some consistency in all this from sitting at Akero's point. And then fourth, quoting, looking for a glimmer of hope for subpart H approval in cirrhotic patients based on summary comments. And I quote, for clinical trials of compensated cirrhosis secondary to NASH, we continue to recommend a traditional approval pathway based on improvement in the clinical benefit outcomes. And then she highlights, DHN is open to discussions with sponsors about other options for approval for this population. So those were hers. Any reactions?
It may be a little easier for the cirrhotic cohort, in fact, to move away from biopsy is the ideal endpoint. Number one, they are already closer to a clinically meaningful outcome on the order of almost 20% at 2 to 3 years, in which case the not only all-cause mortality event rate, but the liver-related mortality event rate may be a little bit more tangible endpoint to measure. And when you think about an invasive endpoint, as we've seen in many trials in cirrhosis, the ability to discern a quantifiable change in liver fibrosis on histology with light microscopy may be very different. Not all fibrosis may be the same. There may be very minor changes or in the even fine changes in fibrosis regression that cannot be readily measured by light microscopy alone, and that introduces alternative platforms, which in and of themselves are surrogate pathology platforms for quantifying collagen and changes using artificial intelligence in liver histology. And when you balance the compensated cirrhosis risk, the risk of bleeding or a bile leak is incrementally higher when we do liver biopsies in patients with cirrhosis than a non-cirrhotic liver. Having done biopsies for almost 25 years of my career, still have a little resonance of biopsying patients with known cirrhosis, because while the risk is low overall, the risk is still slightly higher than in non-cirrhotic livers. So I would be eager to see us as a field move away from biopsy as a quantifiable endpoint as we move closer to alternative meaningful endpoints in cirrhotic cohorts. I think that might be easier than non-cirrhotic.
Louise, do you have 2 or 3 specific things you're looking to see come out of this meeting that you think would be helpful, specific points from where you sit?
I suppose the specific points I'd like to see considered are patient-related outcomes. I think, yes, we have a lot of focus on the histopathological outcomes of these medications, but certainly in the early phase studies and proof of concepts and phase 2 studies, I'd quite like to see some more information coming out about what helps patients. And when Manal was talking about the cirrhotic patients and decompensation and time to those, I think they do make it clear that they're looking at improvement in decompensation. They're looking at improvement in outcomes, but also outcomes for patients earlier in disease pathways are, can they go back to work a little bit longer and more frequently? Do they spend less time integrating with hospital services? There are lots of ways that we can measure the outcomes of some of the clinical interventions that we put in, either with placebo or with the investigative medication. And I think looking for those other markers of people's physical and mental improvement can be actually very informative. And that's what people want. They want a better quality of life. They do want to know that their liver is improving. But which bits improve first? Do you get a better quality of life? Do you get less sort of healthcare acquired costs? There's all sorts of different ways that I'd like to be able to see that these medications are measured by, not just a histological endpoint. So I'd like to see a little bit more detail in there for patient-related outcomes. And I think I'd be concerned if there weren't people raising, like the GLI, about raising the clinical endpoints for patients. And I suppose it would concern me if there aren't any. But I'm sure there will be. I'm sure following the Intercept incident, I think they have learned, and I would hope that they move on from nothing about us without us for the patients, and that their concerns are heard. So I'd certainly like to see some of that detailed a little bit more, sort of bit more meat on the bones, I suppose, about patient outcomes. And I think if they don't, that could be damaging to the way patients want to interact with these studies.
Now, just would it be your read of the document that they are likely to do that, not likely to do that, hard to read one way or another?
Yeah, I have a lot of the confidence in the FDA. I think they are really, really striving to do the right thing by the field. This is a complex disease, multiple moving parts as the science propels itself forward, the drug targets propel themselves forward, and the FDA having the onerous responsibility of ensuring public health and safety. So they have a very difficult job, and I think the more stakeholders are involved and engaged and willing to have difficult discussions around these endpoints and safety and risk tolerance, as well as rethinking how we do clinical trials in this space, this is how we propel things forward. But I am looking forward to Friday's discussion, and I think it will be very enlightening.
Okay, great. Then what I'd like to do is take what I call the third and fourth questions in the document I sent over. Let's wrap them into one, and let's start to wrap it up, okay? And it may be redundant for each of you, but let's try it anyway. Now, the best realistic expectation you have for what comes out of this meeting, and a disappointing outcome that you would consider realistic, but not going to consider realistic, but disappointing. So see if we can put some kind of goalposts around it. Good end, bad end.
I think a realistic outcome that I hope would come out of this meeting and discussion is the ability to look at data in its totality, irrespective of whether it's, quote, surrogate data, because I think we've recognized that biopsy in and of itself is surrogate in predicting an outcome, but to look at the totality of data with unique spectacles based on the targets and their early mechanisms of action. I think what I would be disappointed at is if our experience, particularly as we've come to learn with the evolving phase 2B and now phase 3 treatment landscape, still binds us to a one-size-fits-all and a primary endpoint that's pivotal only on histology. For that, I think I would be somewhat disappointed, because I think we still have a lot of work to do in refining that endpoint in and of itself.
Okay, thanks. Louise, same thing, goalposts. Positive outcome, realistic. Disappointing outcome, also realistic.
I suppose positive outcome, I think, is the emphasis they're putting on the surrogate markers to locate the right patients earlier, which engenders to me that I can get more people involved in clinical trials, more people will be interested, they're going to be less scared, and the outcomes. I think I agree with Manal completely in the one-size-fits-all isn't a good outcome, if that would be the outcome, that it's so rigid in stone, because every patient is different. Every patient has different comorbidities, and I think maybe considering the improvement in the comorbidities as also outcome measures for some of the medical interventions that we're going to put in. So I think that would be a negative. So I agree with Manal on that, if one size fits all is the outcome. But I doubt it. I think reading the documents, there is a lot more emphasis on getting the right patients in, getting those screen failures down by selecting and using the best surrogate markers that we've got at the moment, and improving the surrogate markers. I think improving surrogate markers through early phase trials is a real potential outcome through documentation like this, and the FDA's sort of movement on that. And I think that's a strength. So that's where I'd finish that.
Stephen, would you like to go on to the 2 or 3 endpoints? From your perspective, what is a really good, realistic outcome to this webcast, and what would be a disappointing, realistic outcome to this webcast?
Well, let me just start with what would be a disappointment. The disappointment to me would be just a recapitulation of the white paper that they published in Hepatology. What I would like to see is that be the foundation on which they begin to build the framework that really puts some granular detail to some of these hard questions we have. it's almost like building a house, right? If the white paper is the house, now they're going to come in with the interior design and flesh it out a little bit on the inside. That's what we need. These questions we've raised today are really, really pragmatic questions that helps clear the air as to what needs to be done to cross the finish line successfully in the eyes of the Federal Drug Administration. Without that, we will be left with unanswered questions and ambiguity that will just continue to hamper and hamstring our efforts to find great drugs for patients at the greatest need for help. I would be disappointed if they don't add quite a bit of clarity around these issues we raised today. And I'm confident that they will.
Okay, Stephen, that's great. And for me, I think a lot of this is going to wind up being around numbers. I go back to Kitty's comment about the 1,500 sample and the IHC and where does it have to come from. If we go all the way through 2B without a biopsy and have those patients counted, even if not on the efficacy side of the equation, on the safety side of it, so that we can start to sort out relatively low incidence side effects faster and more efficiently as we go through the process, I think that's a good thing. I think the more patients are required to come through process, and the more they're all required to come through the same process in phase 3, that's a challenge. Now, that's kind of the flip side of one thing you folks were talking about, which is the more heterogeneous we're going to be in the patients we select, and the more confident we want to be that we're learning about the effects on different patients, the more patients we need in trial to do that. So how we balance those two issues, the heterogeneity of patients, the desire to check for low-level side effects, the fact that histopathology is going to be part of phase 3, and creative designs around that. I don't believe we're going to hear how to design it, but I'd like to believe that we will come out of this with enough guidance to understand what designs will and won't be acceptable, and where creative and thoughtful people can make things faster, more efficient, and at the same time more informative. I don't want to see a situation where you've got to trade information for efficiency. I'd like to see if there's a way to capture both in once. As you can hear, we have a lot of high hopes. We have a couple of significant concerns. We'll be back Friday, right after the webcast. The four of us, Kitty, Yale, and one more player to be named later. Everybody have a great week. We will post the hepatology transcript on the website, and we will post a link to the event itself in case you want to get information and try to register. I've not done so. As far as the week goes, have a great time. I want to say thank you before we go to all of our boys, Mike Wilson, who once again is going to have the unenviable task of making this sound great, because the people's comments are great, but I'm hearing some background noise that you'll have to deal with. And Eric Brown, our social media master, Polly Lee, who does our media. Thank you to Stephen. Thank you to Manal. Thank you to Louise for being here with us today. Thanks to all of you for listening, and hopefully you'll all be back with us next week after Friday's meeting. So until then, stay safe, surf on. We'll see you on podcast. Bye-bye now.
You've been listening to the Surfing the NASH Tsunami podcast. Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website. Thanks for listening. See you next week on the podcast.